Okay, so then we will move on to the preclinical drug development field. I think we are now 25 minutes late already, but let's go on. So it's Agnese Compagnone who will speak about preclinical in vivo models. So please.
So we know that is a multifactorial disease of the ocular surface, and it presents a lot of so homeostasis. The loss of homeostasis of the tear film is an important part of the disease, and it presents a lot of ocular signs and symptoms that are very relevant and for the disease. And some of them are tear film instability, hyperosmolarity, the ocular surface inflammation and damage, and the neurosensory abnormalities. So we know that the disease actually very underdiagnosed, and even though it's very underdiagnosed, it's very prevalent. So the prevalence ranges from 17% to 30%, and we know that there are two main subtypes. So the aqueous tear deficient one, in which there's actually a lack of tear production, and the evaporative dry eye, which is the most frequent. So now talking about my project, it's at the moment it's divided into two main aims, and one is the in vivo testing of a new promising ferroptosis inhibitor, and the second part is the development of a customized multiplex ELISA in order to detect dry eye disease biomarkers. So we know that we all know the very important vicious cycle of dry eye disease, and we know that it starts from tear hyperosmolarity, leading then to ocular surface damage, and this leads to inflammation, to the reduced tear quality and secretion, and then the tear instability, and going again to tear hyperosmolarity, which seems one of the main triggers of the disease. But we also know that there are a lot of risk factors in the environment that are related to oxidative stress and to the production of radical oxygens, and also we know that there is the presence of the tear lipid layer on the external part of the tear film. And our main hypothesis is there. Can it be ferroptosis relevant for dry eye? Can ferroptosis be in the context of dry eye? Especially because we have the outermost lipid layer on the ocular surface. We know that ferroptosis is a programmed cell death, and it's related to iron abundance. But we also know that blocking the ferroptosis pathway alleviated the progression of a lot of the disease that are related to ferroptosis. So this can be a very good therapeutic strategies for many disease. And MSCA 3203 has showed therapeutic potential in relevant ferroptosis-driven disease models, but it was never tested in dry eye disease. So this is the main protocol for the preliminary experiment. So the compound was tested on an aqueous tear deficient animal model, and after the surgery there was 1 week of recovery, and then the treatments with the compound were performed twice a day for 24 consecutive days, with the collection of tear fluids and the fluorescein evaluation of the corneal surface. So based on the fluorescein evaluation that was performed according to the Oxford grading scale, we can see that that actually the cyclosporin always has an effect on the ocular surface, and compared to the carrier, which was PBS, because from the stability studies it was shown that the compound was stable in PBS. We can see that the compound has an effect compared to PBS regarding the fluorescent staining. But so this was no, sorry. So yeah, this this was just a preliminary experiment, and the results regarding the fluorescent evaluation seem promising, and the main in vivo experiment is going to be conducted soon. So the protocol, the design of the experiment for the actual in vivo experiment that is programmed in February is slightly different, because we want to check if ferroptosis is relevant in dry eye disease, if there is the presence of this kind of programmed cell death, and we also want to see if it's if the compound is able to reduce the symptoms and signs of the disease in terms of oxidative stress. So based on the safety studies that were performed by Anusha at the University of Finland, the compound will be tested, and it was already tested at a concentration of 100 micromolar, and the tear fluids will be collected in order to check the main inflammatory biomarkers for dry eye disease. Moreover, immunohistochemistry will be conducted in order to check the main genes that are relevant for ferroptosis, and their expression will be evaluated. Glutathione peroxidase is one of the most important biomarker for ferroptosis, and in fact it will be also evaluated through PCR, and the ratio between the oxidized and the reduced glutathione will be evaluated too, and mainly the breakthrough products of the metabolism of the ferroptosis will be evaluated. So, since this is going to be one of the main experiment, a very important experiment, because it needs to test the hypothesis that ferroptosis is involved in dry eye disease, but in the meantime we need to see if the compound is active towards dry eye disease. Feedbacks are more than welcome at the end of the presentation. So now regarding the in vitro, the in vitro studies that I'm performing, so I'm using human corneal and conjunctival epithelial cells, and I'm using hyperosmolar medium at the concentration of 300, 400, and 450 milliosmolar for 6 hours. And oxidative stress is going to be induced through the use of tartar-butyl hydroperoxide at a concentration of 100 micromolar. So the compound is going to be tested at the maximum concentration of 500 nanomolar until 0.68 nanomolar, with the dilutions 1 to 3, with a pre-incubation of 1 hour. And as I was mentioning before, with the in vivo experiment, the main metabolites of ferroptosis are going to be tested, including an immunological panel, in order to see if the hyperosmolar medium is altering and inducing dry eye disease from an immunological point of view. So let's start with the viability. So the viability was checked, and even at 24 hours, the compound doesn't affect the viability of the cells. And the time points tested were at from 2 to 18 hours. And so the compound it doesn't really affect the viability of the cells, of both cells. And after this, the viability in hyperosmolar medium was evaluated at the 3 concentration. And we can see that, so the osmolarity of the medium is 119, while the hyperosmolar media are, as I mentioned before, at 310, 400, and 450. And we can see that the 310 hyperosmolar medium will give us still viable cells, but when we increase the hyperosmolarity, we see that the cells start to lose viability. And in fact, we see that the 450 hyperosmolar medium is one of the main one affecting the viability of the cells. And the time point chosen for the experiment was 6 hours, mostly because I saw that from 14 hours on, there was no viability for all 3 hyperosmolar conditions. So, and starting from the evaluation of radical oxygen species, we can see that the compound, so we can find here the corneal cells in blue and the conjunctival cells in red, and we can see that in the conditions in which the cells were just treated with the compound at the main concentrations, and if we stimulate the cells with the hyperosmolar medium and we treat the cells, we can see that the compound is can actually reduce the baseline ROS levels that was present with just treatment of the compound. And regarding 4H and E, which is one of the main metabolites used, one of the main biomarkers used for ferroptosis, we can clearly see a dose response, mainly for conjunctival cells, and the best responses are at the 116 nanomolars and 55 nanomolars. And after that, we can see that reducing the concentration of the compound, we see an increase of the metabolite of ferroptosis. Then we go to the immunological panel, and we can see that, especially with the 450 hyperosmolar medium, which showed problems with viability, we have an increase in ICAM1, meaning that the reduced viability is affecting the ICAM1 biomarker. And we can also say that it seems that the compound, together with the hyperosmolar medium, is giving a combined effect increasing the concentration of ICAM1. And this effect was also shown on a previous experiment, but with higher concentrations of the compound. Regarding interleukin 17, so it has to be mentioned that the levels detected from the ELISA were in general for interleukin 17 very low, and it seems that the best concentration for reducing the baseline levels of interleukin 17 seems to be 500 nanomolar. But the interpretation of the results, at least for this biomarker, is slightly difficult because it doesn't seem a relevant biomarker for ferroptosis. And regarding interleukin 10, so we can see that the levels of interleukin 10 with the conditions of the hyperosmolar medium is higher, meaning that because we know that interleukin 10 is an anti-inflammatory cytokine, but recently has been showed also having an antioxidant effect. So it means that it increases when there is oxidative stress in the environment, and while with the treatment we see in general a reduced concentration, meaning that the oxidative environment is diminished, so because the compound is reducing the oxidative stress in the environment of the cells. And now we go to the to check another very important biomarker for ferroptosis, which the ratio between the reduced and oxidized glutathione, meaning that if the ratio is increased, and this is the positive control, it means that there is oxidative stress. If the ratio is decreasing, the oxidative stress is reduced. So overall, we can say that the ratio seems to be reduced by the highest concentration of the compound in corneal cells. Regarding the ELISA development, so the other step of the project I was mentioning before, I started developing this ELISA because I needed to include three important biomarkers for dry eye disease in my project. So. And I needed a triplex. So I started performing individual ELISAs at first in order to check the best concentration for every step of the coating. And after that, I decided to go on with the triplex in order to check if there was cross-reactivity among the three antibodies. And so the standard curves from the data were really satisfying, and there was no cross-reactivity among the three. But these are just preliminary data because the analysis was performed with the buffer given by the company. But in order for the validation. Of the method, until now, all the parameters seem satisfied. So the main parameters for validation of the ELISA were tested, and the requirements were satisfied. But there is the need of the because there is the need to use the actual matrix. So for validation, EMA wants you to use the actual matrix that you are going to use for your experiments. So for the final experimental design, I will use tear fluid matrix of just healthy animals, healthy rats, and I will spike them with the antibodies from the company. And then with an experimental design of 7 dilutions, one blank, 2 plates, 2 operators in 2 days, and checking some other parameters for robustness. All the parameters are going to be tested in order to validate the method. And so this is basically the overview of the project, and in the next future I'm going to my secondment to Cologne, and in which I'm going to perform immunohistochemistry and ex vivo experiments. After that I will go on with my experiments, so I will finalize the in vivo research in order to write the research papers that I need, and I will also include a review. And after that, for the very last secondment, I will go to EuDES in order to perform studies on regarding UMC3203, which is the ferroptosis inhibitor, and UMC50, which is a serine protease inhibitor. And these experiments are going to be conducted on a different cell line, which is going to be intestinal cells. So thanks for the attention.
Okay, yeah, there's a question, please.
Yeah, and yes, and very nice work. You've done a lot of
Thanks.
work, a lot of data. I was very interested in your ICAM1 increase.
Yeah.
We've known for a long time that ICAM1 increases, but in your assay, you were actually able to quantitate that.
Sorry, could
you
you?
were actually able to quantitate the increase in your in vitro assay, I believe. Novartis owns Xiidra now, right? Lefitograph.
Yeah.
Yeah. And is there an assay where you can measure interactions between the ICAM1 and the LFA1 on perhaps a T cell?
Yeah, this is going to be very interesting because just assessing ICAM1, it can be relevant, but the actual interaction in terms of LFA1 and ICAM1 might be very, very interesting. I suppose that it can be done with histology colorations, probably, and I can perform it on my in vitro experiments. And because this is very, very relevant, because the blocking the bonding of these two actually decreases a lot inflammation in dry eye disease patients. So what if the compound is acting somehow towards this biomarker 2 and towards this bond 2? It might be very interesting because it's very new. I mean, it's not really we don't know a lot about ferroptosis and dry eye. We are starting to know something about oxidative stress and dry eye, but ferroptosis, this hypothesis might be very, very interesting, I think.
It's important. I mean, this is all involved in trafficking of the cell to the. This is all involved in. Third time's the charm. Yeah. This is all involved in trafficking of the cell into the tissue. I mean, once, if it stays in the vasculature, it's not going to do anything. So that's really important. But you could use that assay even as a quality control thing for what they're manufacturing. You know, with Xiidra, right? So anyway, I just thought that was very interesting. I mean, the data looked very nice.
Yeah, thanks.
Yeah, there is another question.
Thank you. I have one. Thank you for your talk. I was wondering, how do you plan to manage tear collection from rats and to have enough quantity to perform all your ELISA?
The ELISA, because
do you have sufficient material to do it?
So since the tears are going to be collected from healthy animals, so the collection from healthy rats is quite abundant. So I mean, not really much, but I will use I will use 6 animals, and I will collect tears from each animal twice a week for a certain amount of time until I have enough sample.
Okay, it's a pooled collection.
Yeah, exactly. And then because the animals are not going to be treated, because I cannot guarantee, in order for the validation, I cannot guarantee the concentration of the, since it's still the validation procedure, I cannot guarantee the concentration of those biomarkers in diseased animals. So I will collect the tears and I will spike them at several concentrations with the antibodies from the company.
So just, is it a tear washing or a collection with
Yeah,
some capillary or something like this?
Yeah, I already did for other experiments, and that collection was way more difficult because I was removing the exorbital lacrimal glands, so the animals were even producing less amount of tears. But I generally use a microcapillar and a syringe in order to create a little bit of vacuum, so the tears get sucked. But I don't touch. I literally try to avoid touching whatever in the environment of the eye, because otherwise I can have the secretion of
Reflex theory.
yeah, exactly.
Thank you.
I think there was one question there.
Thank you for the nice presentation. I just have a question of, you showed different induction of cells with hyperosmolarity in 6 hour and benzalkonium chloride. Then you also mentioned GPX4 as a biomarker of ferroptosis. I wonder, were you not curious that the expression of GPX also from this in vitro models and to confirm that it's really related to ferroptosis before starting to correlation of cytokines with your molecule?
Could you please repeat
okay.
the question?
Okay, before starting the evaluation, the level of cytokines with hyperosmolarity and
Okay, so you're talking in the in vitro experiments.
Okay, so to confirm ferroptosis, why you were not interested first to investigate the level of like GPX or other ferroptotic biomarkers in these in vitro models?
Yeah, they are included in the design of the experiment. It's just that some experiments were already being performed and some others are still ongoing, and I need to perform them. So, but the GPX4 is going to be tested in the in vitro models.
Okay, so I was asking why you didn't already perform to confirm first the presence of ferroptosis in this in vitro model.
Because the other biomarkers also are
okay, you use just.
I mean, they are all biomarkers for ferroptosis, so they are all used to confirm or corroborate the hypothesis. So, but they have kind of the same relevance, all of them. So.
Okay, so you prefer to use other. Okay, thank you.
Yeah, please.
Sorry, I really enjoyed your talk. I've got lots of experience with collecting tears, diluting tears, spinning off the cells, putting them together, analysing them, correlating them, and I would strongly recommend that you switch to PCR. Tear fluid detection of cytokines is very difficult to do in the short period of time that you have left in your study. Steve Flugfelder, Sencha, they switched to PCR in the tissues years ago because it's much more reliable. There's so little in the tears. It's an absolute nightmare, and you're going to be hounded with contaminants, variability, and these were looking at individual tears. I've never pooled tears except with my healthy controls, where I pool 10 healthy volunteers together. I freeze them in batches and I use each time for the ELISA, typical story. But with the dry eye tears, you're just not going to have enough. They're full of proteases, the cytokines will be destroyed. PCR is really the way I would recommend, but we can chat about it later.
Yeah, thanks.
Also, really along those lines is, if you look into tears, you don't know where it came from. And if you look into PCR, you know exactly which cells are synthesizing it. So it's an advantage.
So, but just to be sure, you're talking about the in vivo experiment, right? Okay, yeah. Because yes, PCR is included in the design of the experiment, but just for checking glutathione peroxidase. But I can improve it with the detection of with the cytokines. Yeah, thanks. This is a very good suggestion. Thanks a lot.
Thank you, Agnese.
Okay,
and,
thanks.
Once again.